It was a gorgeous San Francisco day back in 2018 when I sat down to test a computer vision model I had just trained. I was at my favorite spot in my home office, a little desk that overlooked our back garden that we shared with two other neighbors. And on this particular morning, I was training a model to detect which cells from a cancer patient harbor a dangerous mutation just by looking at those cells under a microscope. Here's an image of those cells from 3 different patients. We've used a handful of fluorescent dyes to tag particular molecules, creating a sort of X-ray that illuminates the cell's internal structure. What you won't see directly is the mutation itself. It's tiny, just 1/100th of a pixel. But this little change in the DNA is thought to drive the growth of the tumor. In fact, if a surgeon removes that tumor and leaves these mutated cells behind, it's possible that the tumor can regrow more aggressive and resistant than ever. Because such a small change in the DNA was having such a big effect on cell behavior, I had a hope. My hope was that it was also creating subtle but consistent changes in the cell's internal structure, and that I could detect those changes visually with computer vision. Now, if you were one of my neighbors in the back garden on this particular morning, you would have looked up into my window and seen something pretty strange. Maybe you would have thought, I'm being attacked by a very aggressive mouse. Maybe I've recently become an enthusiastic football fan. Because I just couldn't seem to stay in my seat. I kept jumping up, sitting myself back down, and a minute later, I'm up again. My legs were no longer under my control, and while this wasn't a medical crisis, it was kind of an intellectual one. The problem was I was getting extraordinary results, and no matter how hard I tried, I couldn't seem to outsmart this model. If this model is as accurate as it appears, we might be able to tell which cancer patients will stay in remission after treatment. We might even be able to find a better way to treat the cancer in the first place. I wanted to run and tell someone what I was seeing, but every time I jumped up, doubt pulled me back down to my chair. I needed to be absolutely sure that I hadn't missed something. This oscillation between excitement and doubt is uncomfortable, but for me, it was a familiar feeling. I'm a medtech entrepreneur straddling the worlds of computers and biology, combining machine learning with a deep understanding of how genes, proteins, and cells come together to make up a patient's well-being. I've invested 20 years in this field, first as an experimental scientist, earning a PhD from Stanford, and then as a data scientist, specializing in industrial applications of computer vision. But when human lives are on the line, biology doesn't care about my credentials or my expertise. That's why every time I was tempted to run and tell someone what I was seeing, I had to force myself to sit back down and take a closer look. I learned this lesson in a very humbling way in 2012. At the time, I was still a graduate student at Stanford, and I had gone home to visit family over a holiday break. One day, we were out to eat at a restaurant, and I was telling my uncle about a recent discovery I had made in my research. I had found a chemical compound that could stimulate neurons to regenerate faster. And with this, I was going to help people recover from traumatic nerve injuries. But just as I was describing the breakthrough in my research, all of a sudden, everyone at the table went silent. And that's when I heard it too. Someone at the table was stuttering because she was having a stroke. And despite everything I had learned about the central nervous system, in that moment, I felt powerless to help her. From that point on, I knew that no matter how many scientific accolades I achieve or discoveries I make, what I know will always be dwarfed by what I have yet to learn. So, how does someone like me accept an utter inability to know the full picture and yet still decide to become a medtech entrepreneur? Well, the answer is that I saw something that, at the time, very few others did. This is an image of cells under a bright field microscope. They're illuminated by a simple white light. While this is probably the easiest way to look at a tumor sample, we don't rely on it very often because it doesn't seem to have much to say. But in the little corner of the scientific world that I come from, we know that this image has so much to say, even if it does so in a whisper. Back then, I had learned to be a little bit careful about telling people what I thought to be contained in this image. But then I'd go to small, focused conferences full of scientists and entrepreneurs who saw it too. When we'd share our data, the energy in the air was electrifying. And at the same time, all of us were watching as compute resources were becoming commoditized and computer vision more and more sophisticated. I knew that soon it would be time to speak up. It all starts with an understanding that the way a cell looks under the microscope is a reflection of its surroundings and its genomic makeup. These two types of input, the external and internal, determine the way a cell behaves and what molecules are expressed from the cell's genes. Traditionally, cancer has been treated with chemotherapy, which exploits a behavioral trait that cancer cells divide rapidly. Unfortunately, this also attacks dividing cells in the rest of the body. So in the last couple of decades, there's been a shift toward precision medicine. Instead of exploiting a behavioral trait of cancer cells, precision medicine targets specific molecules that are abnormally expressed from the cell's genes, or often, in the case of cancer, mutations in those genes. By targeting these so-called biomarkers, the treatment can be both aggressive and at the same time selective for the tumor. The only catch is, in order to match a patient to the right precision medicine, you have to first know which biomarkers are present on the patient's tumor. There are a couple of ways to detect biomarkers, but one of the most direct is to attach a fluorescent molecule directly to the molecule you're targeting. If the cells light up, you know that the patient has a chance of responding. But you can only look for a handful of biomarkers this way before the colors start to bleed into one another. So an alternative is gene sequencing. And with this method, you can look for many biomarkers at once, but you do need a big enough tumor sample to work with, and interpretation of the data can get tricky if the mutations are particularly complex. Regardless of the method, they all require extensive sample processing, access to specialized equipment, and time. It takes days to even more than a week to get results. And because of all of these obstacles, even in the richest countries in the world, fewer than half of cancer patients get access to the testing they need to be matched to these advanced medicines. So let's take another look at this bright field image. I mentioned earlier that I think there's more to it than we give it credit for. Well, it turns out that computer vision can look at a bright field image and generate a synthetic version of a fluorescence image, showing us exactly which biomarkers are present. Each of the colors in this generated image represents a different molecule whose intensity and placement just can't be predicted with the human eye. And because the images are synthetically generated, it opens up the possibility to detect biomarker after biomarker in a matter of seconds. Where biology was once whispering to us, AI just gave it a megaphone. In entrepreneurship, you're expected to be the expert, to speak with authority, and to help others step into your vision. This is a world of action where the motto is often move fast and just break things. Well, it's a complete culture clash when you try to apply that mindset to medicine. The real art lies in being able to move between these two worlds. Action and observation, courage and humility, between making bold statements and asking subtle questions. The reason I leapt up from my chair that day in 2018 was because I was seeing AI decode what I had always believed to be in these images. AI was transforming subtle biological patterns into something we could all see and use to help patients. Today, I'm a co-founder of Moonlight AI, a company that uses computer vision to detect mutations directly from routine imaging of tumor samples. Our goal is to help ensure that patients everywhere get matched to the right medicine faster. And today, when I speak up, I'm far from alone. In the end, innovation has to be a conversation. Yes, it is loud, brash, courageous action, but met with humble and curious observation. It requires knowing when to push forward and when to pause and listen. And for AI to find its place in the clinic, we also need a new model of the entrepreneur. Someone who's willing to boldly put an idea out there and then let data take the lead. So that's where you all come in. Whether you're a scientist, an entrepreneur, a healthcare practitioner, or a healthcare recipient, innovation needs all of us. So the next time you hear a big idea, take a moment to embrace it before you look for caveats. And then ask all of your hard questions. Let's celebrate the innovators who embody that duality of confidence and humility that's required to make a difference in patients' lives. Because these are the people who remember that biology doesn't care about our credentials, our beliefs, or even how hard we've worked. It only asks that we pay attention, and then that we get it right. Thank you.